So again, a very warm welcome everyone to the Kiel Imaging Seminar. It's great to have you. We are very happy to have tonight here Professor Andy Sederman. It's a great pleasure. Thanks for being here. Andy is a professor for magnetic resonance and engineering at the University of Cambridge in the Department of Chemical Engineering and Biotechnology. At the Magnetic Resonance Research Center, the MRRC, his work is focused on developing magnetic resonance techniques for the application to engineering and materials. So a little bit different to our usual topics. He has extensively worked on velocity and transport measurements, focusing on methods to accelerate imaging and speed. His research enhances the study of transient systems through a fast data acquisition and innovative reconstruction techniques that involve, for example, data undersampling. Areas of applications of these methods have focused on single and multi-phase flows, fluid flows in porous media, and reaction and hydrodynamics in multi-phase reactors. Before we start, please keep your questions till the end. You can write them down, you can put them in the chat, or just ask them in person. All right, without any further ado, Andy, the stage is yours, and we are very happy to listen to your research. Thank you.
Thank you very much, Jan. I'm very happy to be invited to be able to come. And thank you for organising this series of seminars that you've been doing for a while now. As you say, this is probably the focus is a little bit different. I think most of them have been sort of on clinical or possibly preclinical areas of interest. And this is more looking at MR and how it can be used in chemical engineering and material science. I'm actually going to be focusing mainly on looking at a specific area of that, chemical reactors, and something of importance to us, looking at them under relevant industrial conditions from one way or another. So this is work, I should say, that I've done with my colleagues Lynn Gladden and Mick Mantle, as well as the number of PhD students and postdocs working with us. And that's at the MRRC here in Cambridge, and within the Department of Chemical Engineering and Biotechnology. So I thought I would start off by saying a few words about why we might want to use MRI in chemical engineering and basically what chemical engineering is actually. So it's an engineering of making things, the processes and the products that we want to make usually on a fairly large scale or an industrial scale. And it's important across a wide range of sectors, obviously making chemicals, chemical process technology, but pharmaceuticals, vaccine manufacturer, food industry, cleaning products, various other sectors that you've got down on the right-hand side there. But some of the things which are sort of common to this, we're often looking at wanting to understand systems which are in opaque environments, often elevated temperatures and pressures, and where non-invasive measurement is typically difficult. What sorts of things are we interested in measuring? Well, we have interest in different chemical species, maybe what chemical reactions are going on. Mass transport is really important as well, because it's not just about doing the reaction, but it's also getting the reactants to where they're going to react, the catalyst usually, and getting the products away from there. So mass transport is very important. And we're often looking at reactions on surfaces, so surface interactions in porous media is very important as well. And so if we look at this is why MR can actually form this unique information-rich measurement technology. So obviously it can look inside in opaque samples, and you can have an environment which has got high temperatures and pressures. And of course, we all know it's non-invasive. That's why it's used so much in clinical medicine. But if you think about looking at chemical species, obviously spectroscopy gives us information about chemistry and chemical reaction. Mass transport, well, we can do flow and diffusion measurements to understand more about those. And actually in relaxation measurements, you can get a lot of information about surface interactions as well. So it provides this really unique measurement technology, which can be used in lots of different ways to help understand chemical engineering systems. So brief outline of what I'm going to be talking about today. I'm going to start by talking about hydrodynamics, turbulent flows in particular. I'll start by talking about it in just a pipe, actually, but we'll then move on to hydrodynamics in packed beds. So I'm largely talking about sort of packed beds today. So what do I mean by that? Well, chemical engineers, we're interested on processes going on at a range of different length scales. So there's stuff going on the surfaces where the reaction's going on, but those surfaces usually will be inside a pellet, a porous material with a high surface area. So we can have reactions on those surfaces with a high surface area. And then those pellets, which may be of millimeter scale, will be put into a reactor, which would be called a packed bed. And the length scales of those may be sort of 10 centimeters or a few centimeters up to a few meters in diameter. So I'll be talking about flows and reactions in packed beds today, sometimes known as trickle beds or fixed beds as well. So I'll concentrate on the hydrodynamics to start with, and then I'll move on to some measurements of reaction. First part, just a couple of slides on pellet scale reaction in a single pellet, and then move on to a bit more information about Fischer-Tropsch catalysis, and I'll explain more about that when we get onto it. But let's think about the hydrodynamics to start with. So most of the systems that we're looking at will, or many of them will be demonstrating some kind of change with time. That may be just changing velocity in terms of if we've got turbulent flow, but it may be also a changing structure with an associated change in velocity as well. And we have two basic approaches to measuring this sort of thing. And the first one is snapshot type imaging, which may be ultra fast. It doesn't have to be ultra fast imaging, but may well be a ultra fast imaging where you need to keep the acquisition time short compared to any system changes. And this gives you an information rich set of data. You can look at all of those fluctuations that you may be having. And I'll look briefly at single and two-phase gas liquid flow in a pipe for that. The other general approach is that of time averaged, where maybe you don't need the extra information, or it's more difficult to get the information, the time resolved information, because the changes are happening too fast. And so the other approach there is just to say that the imaging time must be long compared to the fluctuations, typically with averaging to make sure that you do get a suitable average value out. And for that, I'll be looking at turbulent flows, velocities, and kinetic energies in packed beds. I should say in periodic systems, you can also do triggered acquisitions, gated acquisitions, such as in cardiac imaging, but I won't be talking about those here. So I'm going to look at, this is a few years ago that we did this now, but this was a spin echo EPI-based fast imaging sequence to measure turbulent flow. And this is a fairly sort of straightforward sequence. We've got an excitation part, and then we've got a velocity encode with spin echo, followed by a spatial encode. And that will give you one component of a velocity image. The nice thing is that after that, if your T2 is long enough, you can have a repeat unit and acquire many images after that with subsequent velocity encoding on those. If you change the direction of the velocity encoding, you get for example, three different velocity components. So you completely describe the velocity at each different point in space. In this, we were doing three components in 60 milliseconds. You can also do the same direction, and then you can get acceleration imaging or look at changes in velocity over 8, 16 images or whatever the flow and your gradient system allows. So here are some initial results from those. And we've looked at this at increasing Reynolds number, increasing velocity, but Reynolds number. And typically flow in a pipe, you expect laminar flow below a Reynolds number of about 2000, 2500, and you get a parabolic velocity distribution. No XY flow component, the Z component should be parabolic, which is what you see in, if I get laser pointer up, you can see here and here. As we increase the Reynolds number, you're getting a slight loss of symmetry, but up beyond 3000, 4000, 5000, you can see that we're getting chaotic flow. Now you're getting the turbulence, the turbulent core in the center, and then this boundary layer around the outside, the blue region. And I've sort of highlighted a few what are called bursts or fingers, which are low velocity Z components which also correlate to mass transport from the boundary layer into the turbulent core. So those three components were taking 60 milliseconds. Obviously, we'd like to go faster where possible. And this is, so turns out that spiral imaging is quite suitable for this. It's basic, you can cover K-space in a shorter amount of time for given hardware limitations. It's a very simple MR pulse sequence from the RF point of view. It's just a pulse acquire, basically. But your gradients are a bit more complicated and you need some calibration of those. It's relatively robust to velocity effects as well, which is advantageous. If you've got high velocity, you don't want too much phase being accrued due to the imaging gradients rather than your velocity encode gradients. And the other thing is because we don't have a spin echo here, we can have a short recycle time with a sort of movie type acquisition. Spiral is also very conducive to compressed sensing. So with a variable density spiral here, you can accelerate the acquisition further still. So we've again applied this to the to flow to pipe. We're getting imaging times down here just with a single coil of 5 milliseconds per image, or 5.3 milliseconds per image. And that's for a recycle time. So a frame rate of up close to 200 frames per second, 188 frames per second. Now I have got a movie of this. It's not playing on here, but hopefully you can see that now in an external.
Yeah, works. Nice.
Yeah,
thank you.
Good. So that's being shown at 20 frames per second. Essentially, that's the velocity is the intensity there, and that's being slowed down by a factor of 10 from what that's actually being acquired at, and the real speed of those turbulent velocity fluctuations in there. So that's sort of as fast as we've kind of managed to get with that. An interesting thing that we noticed, and one of the sort of beauties of magnetic resonance in some ways, is so we compared images 80 milliseconds apart here between the EPI version on the left and the spiral version on the right. And the EPI, we said 60 milliseconds we were requiring, and that was quick enough not to see changes in the velocity, so it can be considered snapshot. And that's exactly what you see here. So these are 80 milliseconds apart. These two images look basically the same. The low velocity bursts you can see are in the same place. So that looks good. When we look at the spiral imaging, these 80 milliseconds apart, these look very different. If you look at the low velocity regions, they don't correlate at all between those two images. So it's it's an interesting thing. What's going on here? Is there some sort of contradiction? Is something wrong with the imaging in one case or the other? But it turns out that of course it's perfectly explicable. It's because we're imaging in a different way. So the spin echo SPI that we're doing, we have an excitation at the beginning, and then we measure the velocity of a packet of fluid from that point onwards. And so we're continually imaging the same packet of fluid. It's a Lagrangian type measurement. We're measuring the velocity of a packet of fluid as it moves. The spiral, we're doing repeated excitations for that. And so we're always measuring the same position in space. And so that's an Eulerian change in velocity. And we find that these are give very different velocity autocorrelation function, if you like, from these, if you look at the Eulerian or the Lagrangian frame of reference. And you can see that if I just click on here, we've got these low velocity bursts which are moving. The velocity of the fluid is not changing hugely, but when we consider the same point in space, these are just 10 milliseconds apart, you can see that we have a change in velocity in those regions. So it's just a nice example of how you change your imaging sequence a little bit and you're getting actually different complementary information out from that. But we wanted to move to a multi-phase system. This is a gas liquid system, and with timescales of 5 milliseconds, 5.3 milliseconds for a single image, that's 16 milliseconds for 3 components. That's fast enough now to track not just changing velocities, but the changing structure in a gas bubble rising system, which I've got here. And you can see that we can see the sort of sinusoidal or sinuous path of a bubble as it rises up through the liquid, and we can get the wake shedding. We can look at the vortices information in the wake of the bubble. Now, we thought we'd look at this in a bit more detail. I don't know if any of you have managed to get home and have a glass of something fizzy next to you, but you may be wondering why bubbles are wandering up in the way that they do, and what's the flow features are around there. So we've done this. We've managed to suspend a bubble by having flow going down through the system, and then image the wake of that bubble, which you can see here. Now, if we extract one of the images from that, we can see a couple of interesting things. So we can see that the main sort of counter-rotating vortices in the longitudinal direction. So we've got the bright yellow bits around the outside and the blue bits below the bubble. That's the main counter-rotating vortice. But then there's also a secondary mode of vorticity, which you can see. You can probably see sort of from the arrows in the. So this is in the horizontal plane going round in this direction. Okay. So we can do analysis of that, and we find that the direction of the core wake is seen to be an antiphase to the direction of the bubble secondary motion. So we see that we can so plotted here the motion and the direction of the wake, and we see those are in antiphase to each other, with the secondary vortices reversing direction for every wake shedding event. And this coupling of the bubble path deviations and the wake hydrodynamics had not been quantified before this, and it kind of offers a nice insight into some of the fundamental forces which govern this kind of sinuous path that the bubbles follow as they rise. Right, so I'm going to move on now. So that's looking at flow to pipe essentially. I'm going to move on to some imaging in packed beds. And in this case, we're going to look at time average measurements because the structures are much more complicated. But we want to know about turbulent flows, characterizing them, and importantly for us as well, looking at relevant flow conditions, industrially relevant flow conditions, and sizes as well. So for this is for an application for ethylene oxidation actually, but that means Reynolds numbers of a few thousand, sort of 3 to 6,000, which is in the turbulent regime. So I'm going to I'll go through the sequence a little bit, the method development, the optimization. I'll show a couple of the results. I won't focus on those because that's probably not of most interest to the audience here. But look at how we came up with a robust quantitative imaging sequence to measure these turbulent flows. So the sequence here is on the left. It's relatively simple. It's time averaged, so we don't need to worry about making anything ultra fast. We've actually chosen a single point imaging sequence here with a spin echo. And this is to minimize any of the velocity artifacts. We're not acquiring under the presence of a gradient. We can keep this echo time down to a minimum, so we get small displacements compared to the changes, the structure. So if you're not careful, the molecules will move from a significant distance within the structure over your acquisition time, because we're looking at displacements. We're looking at velocities of meters per second in some cases here. So we've gone for a single point imaging, which provides us with a good, robust, quantitative way of doing this imaging. It's in 3 dimensions. It's taking a while, but we can, because particularly it's 3 dimensions without a read gradient, you can make the most of compressed sensing. So instead of acquiring all the data points, we'd acquire just a subset, around about 15–16% of them. We use a bespoke sampling pattern based on the type of structure that we're expecting to get out from this, which cuts our imaging time. So we're talking about 5 milliseconds before, we're talking about 5 hours now. It's quite a change in order of magnitude, but we actually want to average now. It's important that we average, because otherwise you may not, you may just get a data that is not representative of the full average system. So we do the velocity encode or motion encode with a standard pair of gradients separated by 180. Obviously, as we know, so the coherent motion, we expect to get a phase shift from it. Incoherent motion, from the variance of the displacements, we get signal attenuation. Now, how is that useful for us when we're looking at turbulent flows? Well, we would often think about doing this, it's called a Reynolds decomposition, where we say the velocity at any point in time is equal to the time average velocity plus a time varying component added to that, which is a standard thing that is done in analysis of turbulent flows. Now, how does that compare to our MRI and our velocity measurement here? Well, the average velocity is proportional to our average displacement, and so that, if we do averaging, will just be proportional to the phase shift of our image. What about our temporal fluctuations? Well, the variance of those is going to affect the attenuation, the amount of signal, and so that is going to be related to our loss of signal, log of S1 S0 in our signal acquired. Now, actually, our R squared, average R squared variance term is going to be made up from several different components. It'll be made up from the turbulence, which we want to measure, want to extract, but also from the shear and the molecular displacement. But if we know what the fluid is, we know our molecular displacement, we can estimate the shear from our velocity image, and so we can actually then back calculate out what the R squared term is due to the turbulence itself. Yeah, so time averaged velocity measurement from the phase shift and the variance of the turbulence fluctuations we can get from the signal attenuation. Now, the signal, so that gives us our U squared term or a UX squared term, which is part of the Reynolds stress tensor, which is important for characterizing these systems. It's also used as the closure problem in lots of CFD simulations as well. So this tensor is a important parameter to be able to measure. Now, in here, we're just going to be measuring the diagonal terms here and actually combining those most of the time anyway to get out the turbulent kinetic energy, which is a good characteristic of an important characteristic for turbulent systems. I should say it is possible to get all this, you need slightly better signal to noise, but it is possible to get the off-diagonal terms as well. And if I look at the work of particularly Martin Przewski and Tino in Rostock, Tino Ebers in Sweden, and they've got some really nice measurements of all the different Reynolds stress tensor components. So that can be done, though we're not doing that in this work here. So that's the background. The experimental setup, we stick our packed bed, which is 2 meters long, actually through our magnet here. For a fluid, possibly slightly counterintuitively, we're going to be using gas. We're going to be using SF6. And this is partly because the application is uses gas, and it's a little bit easier to talk to the industrial engineers if you are using a gas when they're thinking about hydrodynamics of the gas, even though water or liquid should be okay. But another interesting thing is if we use SF6 so we're doing this at 6 atmospheres, 6 atmospheres pressure above atmospheric, the kinematic viscosity is actually lower than water, which means we can get to higher Reynolds numbers at lower velocities. And when we're thinking about how far particles move over an acquisition time, it's actually useful to be able to get up to higher Reynolds numbers for these for the lower velocities. It's also, I mean, the SF6 fluorine's got a high gyromagnetic ratio, and we get very favorable relaxation properties. T1s are around about 10 milliseconds, means that we can actually average really quickly for this as well. And we did this for a range of different particle. So these are the structures of the packed beds that I've been talking about. I'm not going to go into any of these different structures, but just to say, I think that again, that is more of interest to other audiences. And I'm not going to focus too much on the results here, other than to say that we can measure these both. So if we see on the left-hand side, we've got a measurement of the turbulent kinetic energy. We did this at 500, about 2,500 and 6,500 Reynolds numbers. So fully over the range of industrially relevant Reynolds numbers. We get the turbulent kinetic energy and the time average velocities as well. I'll show one sort of pair of real results, which I think helps to emphasize why it's important to do these experiments. So on the left here, we've got the turbulent velocity fluctuation variance. So this is the Reynolds stress distributions just of those three diagonal components. And you can see the red and the blue, the X and the Y here pretty much follow the same shape, but the black is actually significantly different. That's the direction of superficial flow. Now, this is in agreement with some PIV observations that have been done on the obviously not of a full 3D bed, but on surfaces. But importantly, actually, many turbulence models, when you're modeling these systems, they assume isotropic turbulence, and we can clearly see that's not the case. So it makes you think more carefully about some of the assumptions that you're making in those models. On the right-hand side, it's interesting. So these are our average, obviously the normalized turbulent kinetic energy, and we find the average value for those is about the same, independent of Reynolds number, though we're seeing a change in shape. So the higher the Reynolds number, the more uniform that distribution becomes. And particularly the lowest Reynolds number, despite people typically thinking that these are fully turbulent, we can see some regions which are not turbulent in this case. And just to hint a bit at what we're sort of looking at the moment, and this is a way to enable some sort of so we always want to be going to higher resolutions and so on, but things get increasingly noisy, and you may want to get shear values, shear rates out, and then you're doing a differential, you get even more noisy systems. So one thing that we're working on in terms of reconstruction now, data reconstruction, is some physics enhanced measurements. And this is using knowledge that the fluid should be following the Navier-Stokes equations and some experimental data, and using a Bayesian framework then within that to optimize the geometry of the system to produce the geometry and flow, which is most likely to come from the experimental data that we are producing. And we can do this in a normal imaging space. You can do this with subsampled data as well. Although that's a little bit still a work in progress. But this is an example. So this is just flow in a contraction. This is the velocity in the direction of the flow, and this is perpendicular to the direction of superficial flow. And we did a high signal to noise image and just a fairly quick low signal to noise, with intentionally low signal to noise NMR image here, and fed this into the physics enhanced optimization. And if we look at the results from the physics enhanced optimization, you can see here it comes out very high signal to noise, very similar to our original, as close as we can get to a sort of ground truth, our high signal to noise images anyway. And when you measure the shear rates within here, even from the high signal to noise data, that's the red line here, you can see that's noisy because you're doing a first differential. If you do a high resolution image, you're going to get some noise within there, whereas the reconstructed image produces some really smooth values for that. Next steps are obviously more complicated systems. We're looking at aorta and clinical or human type systems to look at for that as well. Right, I'm going to move on now to thinking about reaction, and just a couple of slides to start with, very simple on looking at essentially single pellets. It's a single pellet string reactor. So we've got 4 pellets sat on top of each other, and these pellets are porous. They've got a catalyst inside them, and so we're expecting reaction to go on inside. The simple reaction is styrene hydrogenation. So we've got styrene and hydrogen as a feed over palladium on an alumina catalyst, which should form ethyl benzene. Now, the nice thing about this is, firstly, it goes at ambient conditions, so we don't need to worry about temperatures and pressures. But also the reactant is all unsaturated. So if you look at the shifts here, they're all between 5 and 10. The product, we've got this aliphatic region here. So we've got these peaks formed between 0 and 5. And that means whenever we stick in things like porous media, we're going to get line broadening. If you're lucky, it's about a ppm. It may be 2, 3 ppm, depending on your sample. But we can do some chemical shift imaging, some CSI on this, and we get enough resolution of these peaks to get quantitative compositions out. So here are the results. On the left-hand side, you can see this is a render of the blue is where the liquid is. We'll have gas surrounding it, and the grey is the pellet. And we're going to take a slice through this bottom pellet. This is just an intensity image. The pellet is the low intensity. The high intensity is the styrene flowing around the outside. And then we'll have hydrogen flowing around here as well. And on the right-hand side, there are the results from this. So this is now a quantitative map of mole fraction of the product ethyl benzene, in fact. And you can see we've got high concentration around here, certainly heterogeneous distribution, and 3 different pixels extracted out where you can see the growing ethyl benzene or aliphatic part of the ethyl benzene peak, for which we can do the map. Again, something to say why it's important to take these measurements is, if you look at the literature, what you'll typically find is something like this as the expectation for concentration within a pellet like this. And the only axis they have here is R. They assume it's completely radially symmetric, but you can clearly see that that's not the case here. And partial wetting is significantly affecting the distribution of reaction and products in there. And so we're doing some modeling at the moment to try and understand that further. But I'm just going to go on now finally to look at a little bit more complicated system. So this is looking at Fischer-Tropsch synthesis. And Fischer-Tropsch synthesis is the reactants are, it's called syngas. It's a mixture of carbon monoxide and hydrogen. You can get that from a range of different places. It could be natural gas, biomass, could be direct air capture, or CO2 from tailpipe emissions. The hydrogen could then be from electrolysis. And then the syngas, you then in Fischer-Tropsch synthesis, you're producing basically hydrocarbons, liquid hydrocarbons, some heavy waxes. And so this is one of the main hopes for producing synthetic fuel, sustainable aviation fuel. It's one of the main methods for that. And we'd like to know more about what's going on here. We produce hydrocarbons, I said, is mainly we're looking to produce, or mainly we do produce linear alkanes, but there will be some other side products as well. So there might be some branching in there. We'll get some unsaturated products, some oxygenated, some oxygenates in there as well. With the idea being that, you know, normally people would measure what comes out of the bottom of a reactor. It must be beneficial if we can find out what's actually happening in the reaction environment, in the catalyst pores themselves, then we can have better understanding of the system. We can design better catalysts. We can improve the efficiency, the activity, and so on. Generally improve the operation of these reactors. I've got a list of things here which I'm not going to go through, but maybe just, so we had to do a range of different developments, shall we say, to get to the stage where we could actually measure this sensibly in a reacting system. So I'll just skip to sort of a quarter of a slide for each of those, and I won't say much about these at all. But it's important to know so we're producing a lot of water, and where that is, and do we need to worry about that when we're taking our measurements? We can identify, so we have identified at what partial pressures of the water you get formation of water on the surfaces, and then within pores, capillary condensation, and so on. We're also interested in the diffusion of our reactants in our products. We need to know that for being able to understand where we're going to be hydrogen lean or CO lean, and that's going to affect the type of product that we form. So we've done measurements at suitable temperature conditions to directly measure the diffusion of hydrogen and CO in wax and improve the models that were out there in the literature. We want to know something about the product distribution that we're forming. Now, you can think about doing spectroscopy, but we're basically forming different length hydrocarbon chains here. Just, the difference spectroscopically, the difference between decane and non-ane is almost insignificant. It's just basically CH2s with a couple of CH3s at the end. And added to that, then we probably have line widths of a couple of ppm here. There's no way that spectroscopically we're going to get anything very useful out of it, not from a standard 1D spectrum. So what we came up with was something that had been shown before, was that the diffusion within a certain mixture is dependent of the chain length itself. So we're looking at how long whether it's decane or non-ane or dodecane or whatever the different chain lengths. And so the diffusion will be dependent on that chain length. And we've shown with a suitable optimization, novel optimization method, that we can do a diffusion measurement, PFG measurement. If we do an inverse Laplace on that, that gives us a distribution of diffusion coefficients. Now, that distribution of diffusion coefficients contains information about the distribution of carbon chain lengths, carbon numbers. And so from a diffusion distribution, we can extract out a carbon number distribution. We can tell what products we've got in our system. As I'll show later, we get some surprising results out of some of that. So it's nice to have an independent measurement for carbon chain length. And we get that actually through an NMR again, MRI, but a completely different approach. And that's using it's just a nice idea, I think, it's very simple, cozy spectrum. And so along the diagonal here, you see we've got these really broadened lines. This is, let's say, a couple of ppm that we get broadened. The interesting thing about cozy, or about this type of spectroscopy, and cozy here, is that you're looking at J coupling, you're looking at an intramolecular effect, which means you're broadening caused by the local heterogeneities in your field, caused by all the susceptibility differences, is not observed by a single molecule. Different moieties on the same molecule will be experiencing the same local field. And so that coupling is not broadened, and perpendicular to this diagonal, you still retain your spectral resolution. And so just by doing a projection down here, we can extract information about chain lengths, because we see end chains, but also you see peaks for different types of branching as well. Anyway, those are some sort of background work that we did before looking at the actual reaction. And here's a schematic and an image of the reactor setup. So on the right-hand side, here we've got our flow control and the flow coming across the beam here, and then we've got our reactor inside the magnet. And this reactor has to be suitable for looking at operando conditions, real industrial relevant conditions. So this is designed to operate at up to 250 degrees centigrade and 50 atmospheres pressure. And we've got suitable flow control going in, and then we heat the reactor. We have heated separated vessels for wax components, and then a cooler one for the liquid components, and then a GC on the end. The reactor itself it's got steel end caps with a cleverly designed to have a seal with ceramic, even at varying temperatures. Just a little bit of a close-up on the reactor. This are just sort of helpful to look at this to understand some of the later results. We dilute the catalyst to keep it at approximately isothermal conditions, because it's exothermic reaction. So we have 3 layers of catalyst. This ruthenium titania are the catalyst layers, and then between them we have silicon carbide, which is just a diluent essentially, and helps with heat transfer. We've run this for a long time, over 5,000 hours, a few more details there. And to say that, without worrying too much about the results here, we have a GC on the outlet. We can get the standard sort of lab measurements, chemical engineers will be looking at what comes out of the bottom. We can do an analysis on the wax that comes out the bottom. We have a GC on the outlet as well, so we can measure the gas that's coming off. And we can compare with standard literature results. To note as well, we changed the FR here as the feed ratio. So in this series of experiments, we changed the feed ratio. That's the ratio of hydrogen to carbon monoxide in the feed. And I'll show you some results of that in a moment. But to start with, just an image of the distribution of the liquid product within our catalyst bed under operating conditions. This is after 400 hours operation. And you can see we've got those three catalyst layers. We've got this top layer up here, a middle layer, and a bottom layer. We've got some glass wool at the bottom where some of the liquid is pooling after coming down through the reactor. And there we've extracted those three catalyst regions, top, middle, and bottom. And you can clearly see the catalyst pellets here. So the product is essentially in the catalyst pellets. There's not very much that's outside the catalyst pellets. We've just got the product within there. So that's just the MRI, the liquid intensity of the product, where the product is. We can also do diffusion maps of that. And so here we've got the maps of those three different layers. So again, we've got the top layer, the middle layer, and the bottom layer as the three different rows here. You can see three columns here, and these are the three different feed ratios that I talked about before, a feed ratio of 2, 1, and 0.5. And we can see some really interesting differences across here. Most obviously, as we change the feed ratio, go from left to right, the diffusion coefficients are getting really quite a lot slower. So we're going sort of close to 1 times 10 to the minus 9 down to, let's say, 4 times 10 to the minus 10 on the right-hand side. We also, for a given feed ratio, and particularly at the lower feed ratios, if you move down here, you can see we're getting to even lower diffusion coefficients down at the bottom, probably because of some hydrogen becoming more hydrogen lean there, less hydrogen in the system. And there's spatial heterogeneity even within a slice. So you can see different diffusion coefficients across here. But using the inversion that I was talking about before, from the diffusion coefficients, we can extract the carbon chain length from that. So here now is those images where we've done that inversion, and we can look at those in terms of carbon chain lengths. So again, 3 different heights and 3 different feed ratios from left to right. Again, we can see as we decrease the feed ratio, go from left to right, we're getting the carbon numbers increasing significantly. So the purple here is sort of 35 or something, coming up to 55 for the yellow regions. And as we go down the bed, we're getting longer chains being formed again. So down here, we're looking at carbon chain lengths of 60 or 70. You can see the distribution here actually also increases as the feed ratio increases from black to blue to red. Some of these numbers are quite surprising. I'll come on to that in the next slide, actually, but it's worth saying that, so we use the COSY measurement as well, and we get the same average carbon numbers from that measurement. So these are consistent with what the actual reaction environment is. So we are now looking at, this is where the reaction is going on. These are the carbon chain lengths in the reaction environment. So I'll skip that one, just a last results slide here. And I want to, again, sort of emphasize, so the black dots on this plot for the 3 different feed ratios correspond to the measured carbon chain lengths inside the pellets, actually in situ in the reactor under operating conditions. The blue dots are what you catch coming out of the bottom in that wax collection vessel. So if you think what chemical engineers, researchers would do is they collect out what's coming out the bottom, and you'd think that basically what's coming out the bottom, what's in the reactor, is somewhere between what goes in the top and what comes out of the bottom. In fact, usually you'd think it's pretty close to what's coming out of the bottom, actually. So typically, carbon-28, chain length of 28, is used as a representative wax for people to do modeling and try and understand reaction in these systems. And you can see that's pretty much what we're getting out for the collected wax at the bottom. The actual reaction environment, the average can be twice as much or twice as long as that. So you've got really different conditions where the reaction's ongoing compared to what it is at the bottom, and indeed what you think it is when you're trying to understand these systems. Two areas where that obviously makes a difference when you're trying to understand these would be the diffusion coefficient and the solubility of your reactants, your CO and H2 in the wax. So the dashed line here and here as well corresponds to C28. So that's what people would typically use as a representative wax. In fact, we're somewhere within this green region, depending on the feed ratio. And so we have different diffusivities, we have different solubilities, up to 100% different for the solubilities for the hydrogen. So you'll have very different. So your diffusion is much slower than you think it is. Your solubility is much less than you think it is, and you're going to have different ratios of hydrogen and carbon monoxide, which is really important when trying to design what chain length you want to form from this. And so I think this is a really nice place to end, showing the importance of some of these results. So I shall just closing thoughts. I think MRI is really powerful for engineering as well as clearly clinical applications. And there's a whole wealth of more information that needs to be found out in packed beds for heat mass transport and for reactions. And I'm sure there are lots of other areas to look at as well. And I should finish by thanking Lynn and Mick again, but my students, Scott Elsgersmer in particular, did a lot of the packed bed work. The next line, Andy, Alex, and Dan were involved in the single and two-phase flow, velocity imaging, and Qingwan, etc., were involved in the reaction work. Thank you very much. Thank you very much for listening.
All right. Indeed, thank you so much. Very, very much impressive and fascinating work. What you can all do with MRI.
Yes, lots of different things that you can do with it's an incredibly flexible, wonderful world of things to play with. Yeah.
You can probe so many mechanisms, and from those results you can derive then physical parameters like chain length. It's very cool. I once used spectroscopy and pieces of rat livers to estimate the chain length of the fat molecules. Right?
There we go. Yes.
I invite the audience for questions. And while the audience collects courage to ask, I'll ask. So the latest question I wrote down some here. The latest question I wrote down is maybe the easiest or the most stupid one. If you go to the last slides where you nicely showed the data on the pellets, why? One more, please. The images of the pellets where you see actually, yes, here. So I'm not an expert of the Fischer-Tropsch reaction, but why do we see the surface of the pellets mostly?
Sorry, why do you see
we see the surface of the pellets, right? Or what do we see here? Why don't we see the space in between?
These are values for inside the pellets. The brightness is to do with the brightness of the intensity. So there's the color scale corresponds to the chain length, and the brightness corresponds to essentially how much signal we had in the image.
So, and why don't we see signal in between the pellets?
Because there's no liquid there. So that space is filled with some void space and some silicon carbide, which is this a diluent. It helps helps heat transport in there. But essentially, you're producing this liquid, and as the reaction progresses, you're producing more. It's being forced out of the pellet, and then three things are happening to it when it comes to the outside of the pellet. One is it's vaporizing. Right, so we have a range of different chain lengths, and those below sort of C10 will largely vaporize, depending on the partial pressure of those within the system. You'll have some, particularly the shorter chains in there, will be flowing straight out. So they'll be just under gravity. They will be falling to the bottom of the reactor, and that's where we collect our product. Interestingly, we also have some, and probably the longer chains will be there is some liquid which is retained outside the particles, outside the pellets. Now, we don't see a lot of it here. It tends to build up over time. And we're doing further experiments just at the moment, actually, to look at the effect of that on the mass transport, because usually when people try and understand these things, they think about the mass transport from the surface into the pellet. But if you start to get that void space filled with liquid as well, then you need to think about mass transport through that liquid outside the pellets and then into the pellets themselves. Yeah, you get interesting sort of hidden reaction kinetics. People are interested in kinetics. What happens if you double the temperature and so on, and you get hidden reaction kinetics hidden by mass transport. So a chemist might say, well, you just need to know what the activation energy is, and then you can work that out. But actually, the mass transport becomes key in trying to understand how these systems are going to operate.
So are these white voids filled with something, or with matter the silicon carbide, or they're just voids and where the liquid can go into?
So they are filled with silicon carbide particles, which themselves have voids.
Okay. So the liquid can go through.
The silicon carbide particles are solid. They don't have voids, but there are voids between them. And so there's space for gas and liquid to flow through.
Yeah, and I can see that most of it evaporates quickly, and so then you will have less density and less signal.
I think most of it probably is liquid, so it's flowing down, but some of it will vaporize, some of it will be liquid, but most of it will flow through there. And it's probably the longer chains. I mean, what's really interesting, I think, is what's coming out the bottom is C28. Where it's reacting is C50 or something. So you must have an interesting different mass transport, different transport mechanism for some of these longer chains. They must be diffusing out, flowing out more slowly than what's coming out the bottom.
Yeah.
For the mass balance all to work out.
It's really cool. I like it. That's cool, right? And I agree with you as well that on many different topics, it's good to look at what's actually going on.
Yes.
It's a little bit off topic, but for the microbiome, same thing, right? You take stool samples and you want to understand what the microbiome is, but we try to do it with MR to see actually what happens in the bioreactors.
Yes, absolutely. Yes, no, that's getting an insight into what's actually happening, right? If you're sampling stuff, you're taking it from a particular place, you're taking it at a particular point in time, and it may be representative, but it may not be.
Thomas Kampf, hi Thomas, is asking a question. Hi, really interesting talk. Maybe I missed it, but how do you handle through slice flow and the flow encoding multi-spin echo EPI sequence?
So this was the idea of this is in the multi-slice. So it's not multi-slice, it's single slice excitation, and then multiple refocus of the fluid that was originally in that slice.
Yeah. And I could guess that the refocusing is then non-slice selective, but global?
Exactly. The refocusing is non-slice selective. And so there is a limited time over which you can do this, because you're still doing the spatial encode in the XY plane every image. And so if you have very high X or Y velocities, then you will get fluid that begins to migrate from one pixel to another. And you can see a bit of that happening, but actually, what the velocity is, you can calculate how significant effect that is. And typically we, as I say, over sort of 100 milliseconds, this was not significant. And this difference between the Lagrangian and Eulerian, monitoring a packet compared to monitoring a position in space it's just another really nice example of how flexible MRI can be. They're both right. But they're giving you different.
Just show different. Yes. I had as well. My first question was to that slice or to that data, but I think you answered it. So you do one excitation, then multiple echoes for multiple images, right?
Yeah, that's for the first one.
Yes.
Yes, yes.
Yeah. Cool.
Um, and
Thomas,
yes, you can
sorry.
change the direction of your velocity encode. So you can turn it into an acceleration imaging sequence as well. You can have a zero first moment to it and a non-zero
Mhm.
second moment, and so you're actually doing acceleration imaging. And you with an echo with a train, you can get a velocity autocorrelation function from that, and compare that to a velocity autocorrelation function that you get from the spiral. I mean, this is not inherently a difference between the spiral and a rectilinear EPI, right? It's because we have signal excitation in different ways in the different sequences that we're using.
Yes, exactly. You basically have a long time between the excitation and the encoding, right, with the spin echoes.
Yeah,
And the FID style sequences, whether it's spiral or whatever, it could be EPI as well, I guess.
yeah.
You just have very short time between excitation and encoding, and there you have echo times in between.
So the spiral works out. It's better for shorter T2 star systems, particularly if you're. I mean, EPI is a little bit more difficult in my
Uh,
experience. Anyway, I'm sure
mm,
others have got it off to a T, but it's a little bit more difficult if you want to do a sort of center out type EPI, whereas it just comes naturally from the spiral. Uh,
yeah.
and so where we tend to have slightly shorter T2 stars than probably many clinical systems, particularly because we're working at higher field strengths as well. I didn't talk about field strengths. We're typically working at 200 or 300 megahertz. So
Mm.
um,
4.7
even
Tesla, 7
400
Tesla,
megahertz. So
mm,
yeah, up to nine Tesla.
9.4. Yeah.
Um, and yeah, so the spiral it can be quick enough, and it is less susceptible to. You always have to consider the first moment of your imaging gradients, right, and see what effect those are having on your phase
Mhm.
due to flow, because you want to make sure that your phase shift is that you're measuring is due to the flow, or rather than because of, or you want to separate that out from your imaging gradients.
Thomas is asking a follow-up question. So one observes the only so one observes the only the in-plane components. Do you expect that the non-observed third dimension can become a source of drain of energy if flow is converted from in-plane through plane and vice versa?
So we do measure all three components. In both cases, so this is as you were saying, because all the other pulses after, well, we have the two different the spin echo and the spiral. The spin echo, that 180-degree pulse um, uh, is a broadband refocusing pulse, and so it doesn't matter that the fluid is moving out of the slice. What's Yeah, so we can still monitor that as it moves through our system. It is important to check that within the time frames that we're doing the measurement, that we don't have submitters too much motion in the XY plane, but actually it doesn't matter how far it moves in the Z direction, in the direction of the superficial flow, because we don't have spatial resolution in that direction. Um, yeah, so we do get that information um, from the, and obviously the imaging itself doesn't affect the flow in any way.
Thomas, you can speak up if you like as well, like by microphone. Maybe that's easier if you want to discuss. No microphone. All right. Um, so I think your first question was the through slice flow, and I think that is easily explained because the 180 degrees is non-slice selective, so it doesn't matter. You fully record the through slice flow,
Yeah.
and then you just said you measure all three components of the velocity, but I think you measure them one at a time, right? You don't measure them
Exactly.
at once.
Yes, we're
Yes.
always measuring them one at a time here. And so it does, for the idea of a snapshot, you need to ensure that time is short compared to the changes.
Yeah.
Um, but one of the nice things that, so if you turn your gradients to all be in the same direction, you can actually monitor exactly how much those are changing as a function of time. And as I Yeah, what was it? I
Um,
can't
I,
remember exactly which slide
yeah,
it was, but
I have a question there too. First I thought, how can you measure turbulences if you need to average signal, if you need sinus style, but you don't do sinus style, you do snapshot. I understand that, but you can also one snapshot is only one velocity component, right?
uh, one snapshot is one velocity. Well, no, I mean, I think I would argue that three components are still one snapshot. So it takes three times as long,
Sure.
but the requirement is that time is short compared to the changes.
Yeah. Okay, I can see
Therefore, it
that. Yes.
becomes snapshot.
Yeah.
But they
And I mean five
absolutely are.
milliseconds, right?
So in the faster case, it's 15 milliseconds, 16 milliseconds for the two-dimensional image for three components, which
Yeah.
is slow compared to optical imaging, and it depends what you're comparing against. But what's really important is thinking about the system that you're looking at and whether that's short enough for
Yeah.
your application.
Yeah. It's super fast. For me, it's super fast.
I think one of the advantages of not having anything remotely clinical is that there are nobody puts any restrictions on what you can try and do.
Hmm.
So there's no slew rate, maximum slew rate. You just try it, and you measure what's happening actually, rather than keeping it necessarily below a certain value. You push it, and then you measure what your trajectory is, rather
Yeah.
than worrying too much about where it should be.
Yeah. Yeah. It's good. And Thomas says as well that that's good. And now he sees and he says again, it's really nice work.
Thank you very much, Thomas.
So please, audience, we are overdue with time, but you have the last chance to answer a question. You can speak up or write it in the chat. Otherwise, we will leave Andy to his well-deserved evening and everybody else too. Thank you so much, Andy, for being with us tonight. That was
Well, I
fantastic.
really need to thank everybody for joining. I know it's even later for you than it is
Yeah.
for me. So thank you very much for sparing an hour of your time.
Yeah, it was well spent. It was well spent.
Thank you very
Thank
much.
you so much. And thanks everyone for joining us tonight. And everyone, have a great day.
Thank you very much.